Hey guys, so I wanted to get this recording to you for chapter 45, since we are missing that in class. So with chapter 45, we are talking about population community ecology. We're also talking about animal behavior. They've snuck that in at the end. So we got a lot of things to cover. So I'm going to try and get through everything. I'm not going to do the videos or anything like we normally would in class, but I do encourage you to please check those links out. And look at all those guys because there's some good stuff in there. There's good stuff in there. If you hear my dog snoring in the background, I apologize in advance. She is a really loud snorer. All right, let's get started. Okay, so it's talking about understanding population ecology is super important. We want to know when we are studying things, how much of the animal there is out there. Are there other animals that are impacting it? What is their population? Are they impacting it in a way that they are overcrowding and taking other resources of the animal that we're trying to study? So maybe we're trying to study a specific endemic type of fish, and then we've got these carp that are here and as an invasive species and they're taking over. So they have an example of how people are going in and sampling, shocking the electrofish survey, and shocking them to count them. And then there's another example of the picture in the picture where they've poisoned them um, to study the population. But studying populations is absolutely key, important in biology. If you are going to study an animal, you want to know how many of the animals there are. All right, so we will talk about demography, the study of population. If you were looking at the number of individuals, it's going to be your population size. But if you want to know individuals per area, per unit area, then you're talking about density. And both of those are important too, because density is going to matter when we talk about population and fecundity and that kind of stuff. So for example, I pulled up Atlanta, and according to Wiki in 2024, it's the city of Atlanta is under 500,000 people in there, 498,000. And that is, again, just the city of Atlanta. But if you include the metro area, we are at 5.6 million people in the metro area. Um, the density is not too bad. It's not too bad when you compare it to something like New York, because right now Atlanta is the ninth largest city. But if you wanted to compare it to New York, which is very large, they have 7.9 million people, and the density is crazy. I'm sure everyone knows that housing is really difficult to find in New York and outrageously expensive, and a lot of people are living in very, very tiny, small places. So the density there for people is 26,000 people, 397 people per square mile, 26,000 people in a square mile in 2024 in New York City. So that's just an example as comparison. We are talking about population density versus body size. So if you look at the bottom on the x-axis, I don't know if my mouse shows up when I'm recording, but I hope it does. On the x-axis, it's the mass, how big the animal is, and on the y-axis, you've got the density. So in general, the bigger the animal is, the less there are per area. That kind of makes sense, right? Areas can only support so many creatures if the creatures are gigantic. One method of sampling that you might want to do if you were studying animals, you might also want to study the vegetation in their habitat. So one method that you could do to study and sample vegetation is a quadrat sampling. We have some of these on campus too. I've given them to my wildlife students to go and sample vegetation in the different habitats. So in this one, we've got a quadrat. It looks like a meter squared about quadrat sample, and you can see the little squares inside of there. So with the little squares inside, you can sample different species within the squares and give like a percentages. So like 30% of the squares are or this specific species of grass, and this other kind is a clover, and this square over here, 1/16 of the squares is this species. So you could do percentage, or you could also do like all of the plants found in that one square and then go to a different sampling with your quadrat, and you'd randomly sample your area, and then you calculate up the mean, and that would tell you about the population of the vegetation of a specific species or percentages of species in your area. Another type of population sampling is mark and recapture. Don't know if it's going to let me move this out of the way, but I really want to put this on there. This is one that I like to play with on the board, and I did not hook up my Apple Pencil to the situation today, so I tried to type it in. But on mark and recapture, this is something that's used in wildlife all the time. And so you've got a population of animals and you want to know how many there are. And so sometimes you will be able to get your hot hands on to mark an animal. Sometimes you don't have to mark them. You can capture them with a picture, a game cam or something like that, and they got natural markings and you can identify them that way. Maybe they're stripes if they're tigers or zebras or something like that, or whale fins. You could identify them by their natural markings. But if not, you could mark them. You can mark them. And so, if you wanted to do population samplings based on those marks and recaptures, then you would do this example over here that I've got in the corner. So N is our population number, and the number of marked, it would be up top, and then we're going to multiply it by the number that we captured on the second event. So you got one trapping event where we marked everybody. And then we got a second trapping event, and we're going to capture everybody. And out of that total number that we captured, we're going to put that up here in the second location. So the number of animals we marked in the first event times the total number of animals that we captured in the second event. And then we're going to take that number and we're going to divide it by how many in that second group were marked from our first event. So in this example, we've got, I'm going to say it's something small. Mice. We're going to pretend it's mice. In a big old field, a huge field, several acres. This whole valley. We're going to capture mice in this whole valley, and we're going to do these random samplings. So we're going to capture mice. And in this first trapping event, of all the mice in the very first event, we captured 80 mice. We captured 80 mice on the first one, and we marked them however we wanted to mark them, hopefully humanely. We marked them. And then we're going to come back in another trapping event, maybe in a month. Who knows? Maybe it's in 6 months, maybe it's a year, but we're going to come back later in a second trapping event. And in the second trapping event, instead of 80 mice, we capture 100 total mice in the second trapping event. So we do 80 times 100, that's going to give us 800. No, 8,000. It's going to give us 8,000. We're going to put that on top. I'm going to put that on top. But remember, we had to divide it. So when we divide it by the number of marked ones that we found in the second trapping event. So out of the initial 80, only 20 of them out of the 100 in the second trapping event were caught again. Only 20 of them were caught again. So out of the 100 that we caught on the second trapping event, 20 of them were marked that we had caught previously. So we're going to put 20 at the bottom. So we're going to do 8,000 divided by 20, and that's going to give us 400 as our population estimate of mice in this entire valley. So we're going to estimate that 400 mice live in that area based on our initial mark and then our recapture event, and comparing those two numbers will give us a mark and recapture sample. So that's one way to mark to estimate population is mark and recapture. And you can see all the different ways that the animals are marked. We've got the bighorn sheep, and they've got radiotelemetry collars on them. We've got a California condor, and it's got these wing bands that go across, and they'll have numbers on them to individually identify them, just like the radiotelemetry collars individually identified those guys. And then you can see the tag on the dorsal fin of this fish. What type of fish is he? He's a salmon. In C over here. And then another cool thing, instead of radiotelemetry collars, sometimes we have GPS collars now, which are replacing the radiotelemetry because GPS is so much better. And the more the technology evolves, the lighter the equipment gets and the smaller it gets. So it's way easier to use them on animals. So we have been using them on several animals. Those collars in the A letter on the bighorn sheep, those might actually be GPS collars and not radiotelemetry. But if you click that link, the shark tracker in the bottom, I don't know if y'all checked that out before, but it'll link you to a website where you can track sharks and different types of sharks. There's great white sharks, and I think there's some tiger sharks on there. But it's kind of cool where it will show when they, when they surface, it'll ping the satellite, and you can sort of track where they are in sort of kind of real time, which I thought was cool. So this is an example of mark, all the different types of markings that we got on this slide, and then recapture and how you could use that to estimate populations. Is it gonna let me keep on moving? There we go. All right, so another example of mark and recapture are this is my feral cat experiment from my master's program at Auburn. And so with our feral cats, we were trying to, for my thesis, we were trying to see the impact of trap, neuter, return versus the traditional method that was going on with animal control, where they would just come in, they capture all the cats, they kill them, and euthanize everybody. And then the theory prevailing of the day was that created a vacuum and other cats would move in So the more humane method was trap, neuter, return the cats to the location. So that's what I was studying for my master's. And so we had the feral cats and we had 7 different colonies. And my husband and I built these cat houses here. And inside the cat houses was food and water to attract them in there. And then there was a game cam. So we would take pictures of the cats every time they went to the feeder. And so I would use their natural markings or their radio telemetry collar that I put on them, and I could mark and recapture them every time they came and got on the camera. I could also go out with my radio telemetry that I did every 3 days and GPS locate their points with a handheld GPS back in the day, which was less fun than possible little, air tags that we could probably put on them right now. But this was 20 years ago, so this is old tech. But you can also see from a distance another way that you could mark a cat that's been trapped, neutered, returned. If you look in the cat picture on the upper right-hand corner, you can see that his ear is tipped. His left ear is tipped. So that's traditionally a sign of trap, neuter, return. So if you ever see feral cats, like oftentimes I see them behind the dumpsters of like restaurants and stuff like that, is usually where I might see them around my personal area. And sometimes somebody is maintaining that colony. There's a colony caretaker and they've trapped, neuter, returned those cats, and you'll see their ears are tipped. So if you see that where it's tipped off, that's a signal that that cat has been trapped, neutered, and returned. Species distribution. So we've got 3 different patterns of species distribution. We've got uniform, and you can see the little dots are relatively spaced out, just like it sounds, uniformly. Then we've got clumped, which is more herd-like, where everybody's together. They are not spaced out, utilizing the space like it is in uniform. And then we've got completely random, and our example there is daffodils, which are blooming right now. And you can see that, they literally just go where the wind blows, they land where they land, and it is absolutely random distribution. When we talk about species distribution, we also like to talk about, I'm going to make sure we get through all my boldface terms, life tables, and then we'll talk about survivorship curves. So on the life tables, I can't remember if this was in the textbook or I stole this from another class, but we've got the Dog Mountain sheep here in this life table. And if you look on the far left column, you can see this cohort of animals. And so they're going to track them in number of years. And then you can see like 0 to half a year. So first 6 months, where's my mouse? There it is. First 6 months, 6 months to a year, then 1 to 2, 2 to 3, on and on and on until about age 14. And this cohort had 1,000 little sheep in this cohort starting off. And in the first 6 months, 54 of them died. And so 54 out of 1,000 died off. You can see the mortality rate per 1,000 is 54 exactly, because we are in the very first step of this life table. In the 6 months to 1 year, you can see that that was pretty much one of the hardest times for these little baby sheep, was 6 months to a year, because 145 of the little babies died, and that left us with 946 out of the original 1,000 at the end of 1 year. And you can see the mortality rate there was 153 out of 1,000. Then at 1 to 2 years, it seems like things are getting better. We're a yearling now. We're getting our feet under us, and so mortality rate at that stage was 12. And it was better. So we got 12 out of 801. Mortality rate is only 15. It's only 15 out of 1,000. So that's a much better mortality rate when it looks like for all of the rest of the timetables. And then as they reach older age, adult age, and aging, more elderly, senior aged sheep, it looks like mortality rate starts to double and then double and then double. And then you can see after age 7, things are not looking so great for the sheep. And then the oldest last sheep was age 13, 14 out of the original 1,000. So this is an example that you might see if you were trying to study a species like this and determine its mortality rate, study that population, maybe manage an area for a specific population. You might see these life tables. Survivorship curves are also things that are talked about in biology all the time in ecology. So we've got 3 types of survivorship curves. There are type 1s, and that's us. So if you look at the x-axis, that's your life expectancy. How long are you expected to live? So the x-axis is how long you're expected to live. And then the y-axis is going to be the number of individuals there. So we all start off at the very top of the y. And then how does it go? How many are going to reach maximum life expectancy? So with people, Now, traditionally, with modern medicine and agriculture and all that good stuff, traditionally people are going to make it to old age. And then you can see the blue line tapers off and it goes down. So we are type 1. We typically make it to adulthood, and then old age. Hopefully, we make it to a ripe old age. Versus type 3. Type 3 example is trees. Remember we talked about how the dandelions just blow their seed out wherever, they're wind distributed. Trees, we talk about how trees are also gonna get their pollen out there, and hopefully it lands in the proper spot, and hopefully that cone releases its seeds and that gets blown by the wind and it lands someplace good. But the majority of the seeds don't land in places that are great when they sprout, and they don't make it to adulthood. So if you look, you can see the green line for the trees, which is the type 3 curve, drops off steeply to make it to the first year, the second year. Odds are small, but once you've made it there, it looks good. It looks good for you. But the beginning stages of life for type 3 species are rough, are really rough. And then in the middle, you can see type 2. Type 2 example is birds. And birds don't have great odds no matter the timeline. If they're early, nestlings versus teenagers versus adults, it's still about a 50-50 shot for a type 2 species. For a type 2 species, you got a 50-50 shot of making it to the next year every year. So definitely know the type 1, 2, and 3 survivorship curves and examples of those, because I totally have a test question about that one. But we'll go over test review later. All right, so life histories and natural selection. Life history is going to be important. We need to know what is happening over something's lifetime and how does that impact it. Energy budget. This is when we talk about having to budget everyday energy, the life of just getting through the day. How much energy are you spending on that versus reproduction, and how much energy is spent on that, and parental care. Um, it 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 matters. It's an expense. Another term up here is fecundity, and fecundity is going to be fertility. It's going to be fertility. How many offspring could this species create in different circumstances, different ages, different environments? How fertile? What is the fecundity level of that organism? Early versus late reproduction. Absolutely pros and cons. Pros and cons for both. Um, and usually I like to pitch this to the class and get y'all's input on it. But for early reproduction, one benefit would be that I'm speaking to this as people, as humans, and early reproduction. A benefit would be that if you had offspring early-ish, you're still young. You're still able to care for those children well into those children's adulthood. Um, a con of early reproduction, speaking as humans, if you are very early in reproducing, this is terrible to think about, but it's happening. If a young girl was forced to reproduce at like 11, her body would stop growing. Her body would stop growing, because at 11 you're not fully developed, right? You're still developing. But if that happened, your body would stop developing, because your body is focused on creating that next life. And your development would stop. And that would absolutely take a toll on your body, if you were further along, maybe 18, 19, 20, your body is fully developed. Your body is fully developed. If you're a person, you may or may not have the financial resources at 18, then 20 or 22 or 25 or 30 or 35. So pros and cons. Pros and cons. There's pros and cons on early. There's pros and cons on late reproduction. If you were late reproduction as a person, um, you might have those financial resources that we spoke about before that you might not have when you were early on in your career. You might have more financial resources. A con, um, you may wait too late. You may wait too late. And the fecundity, fertility level drops off for women as we age. Men are, we talked about in meiosis, men are creating sperm all day, every day. But we talked about how egg development shrinks down, and then chances of non-disjunction start to skyrocket. And you, I think at the age of 35 is considered advanced maternal age at 35, which does not seem very old to me. But that is considered advanced maternal age. What if you waited too late? And now there's technology, you can freeze your eggs and all that good stuff. But back in the day, just think about pros and cons. So a pro, you might be more established and have more resources for that offspring. A con, you, if you wait very long, you might not be able to enjoy and have the physical fitness that you would have with a small toddler, if you were younger than you were, if say I had a newborn right now at age 46. I know my fitness level is not the same as it was when I was 26. Me personally. So pros and cons, early versus late reproduction. Early can absolutely take a toll on the body. Late can also take a toll on the body. There's pros and cons. I could go on a whole diatribe. I'm gonna stop now. Um, the last stuff on here is single versus multiple reproductive events. So there are different animals that may exhibit similarity, and with that one, that is where you only have one big event in your lifetime, and that is where you are going to get together, reproduce, pass on your genes, and that's it. That's it. Just one single event. So I have this link over here to the corpse flower, and I've seen several videos now of people. They must have planted this. This must have been like a really big thing 30 years ago to plant, or 20 years ago. But the quartz flower, a lot of them I've been seeing in the news are blooming, and they have similarity. They bloom one time and that's it. And then they have all their offspring, and that's it. Salmon, we discussed salmon too, same thing. Multiple reproductive events, iteroparity. So maybe you reproduce one time a year, but it's every year. So you'll have seasonal estrus, like deer do. They'll go into estrus, it's reproductive season. It's time to do that. It's just this one specific time of year, and then you wait till the next year. Or you're like people, and people's reproductive events are not necessarily tied to intercourse, is not always tied to producing babies. It's not one leading to the other. Like in animals, it might be. So we've got our different examples here. Yeah, we've got the salmon, we've got antelope. So salmon is our similarity, one-time event. And then we've got iteroparity, where we've got multiple times, but just one time a year. But as long as you are making it to the next year, hopefully you'll make it to reproduce again the next year. And then we've got examples of chimps, just like us, that intercourse can happen any day, anytime. And it is not specifically timed to ovulation. Like it doesn't have to be timed to ovulation. And then I got a link to the mating game, which has it's the full video, it's the full movie of it. But it's got all sorts of really cool footage. I can't remember if it's Nat Geo or PBS. I don't remember who produced it. But it's got really cool footage of different types of courtship behavior between all different types of animals. So it's really cool if you wanted to check it out. Heads up, it is like an hour long. So just letting you know. Okay, we talked about fecundity. Remember we talked about fecundity and it was fertility. So this is just an example with fruit flies. It is getting warmer, and we are all going to have fruit in our kitchen, and some of us might get fruit flies. These little guys uh, are really great for experimentation. A lot of people in science experiment on fruit flies and genetics and all sorts of stuff. But in this specific one, they were testing fecundity and the male's sperm. So if the male had lots of sperm, then they would go for whatever female they could get a hold of. and reproduce with. But if they were sperm depleted, you can see that they would go for it's a 15-to-1 or a 3-to-1 ratio, that they would try and choose a larger female. So we are assuming that the larger females means that they have more fecundity. They are going to be able to produce more offspring. So if the sperm were depleted, if the male had a low sperm count, then that male would choose to mate with a larger female versus a smaller female. There we go. Okay. Population growth. So curves. Definitely have a question about curves. So we've got exponential growth. So it is a J curve. We call it the J curve, exponential growth. So you got population growth over time, and you can see it just skyrockets exponential growth. That is normally only happening like in a lab in certain type of situations, because eventually you are going to hit over here this logistical growth, which is called an S curve. You're going to hit a carrying capacity. So the red dotted line is carrying capacity. So whatever your organism is, maybe it doesn't have enough nutrients, doesn't have enough food. Maybe it's running out of water. Maybe it's a wild animal and it needs enough shelter. Maybe it doesn't have a place, have a mate to reproduce with, and it's running out of trying to pair up. There's some sort of limiting factor there that is going to limit the population growth, and it's called the carrying capacity. Whatever that factor is, it's going to eventually cause that J and exponential growth to curve into an S shape when we hit carrying capacity. So we got exponential versus logarithmic, also called J curve versus S curve. Yeah, lab growth, or we can, input new nutrients in and get rid of waste products all the time. We can see absolutely exponential growth in a lab type setting, where before, down below, we've got this S-shaped carrying capacity with what type of seals are these? Does it say? It does not say what type of seals they are. They are super cute. I don't know. That's not a leopard seal. I don't know what he is, but he's cute. So we got these cutie patootie little seals. And back in the day, humans did what humans did, and we killed off lots of them. And then once we made that illegal, you can see 1975, I'm assuming the law went in place around that time. And once it was made illegal that we could no longer hunt and harvest these for meat and fur, you can see this J-shaped growth, crazy growth until about 1990, where they eventually ran into carrying capacity. So they've gotten into all the habitats that are great for the seal, and they have hit carrying capacity. Maybe food is limited, maybe some sort of resource is limited for them, and they hit the red dotted line at carrying capacity, and that's about where they are at about 7,500. All right, population dynamics and regulation. Fecundity versus population density. Remember we talked about if the fruit flies were sperm depleted and they only had so much left, they were going to choose what they picked was the more fertile female, the larger fruit fly. In this situation, fecundity is related to population density. So on our x-axis in this diagram, we have the number of worms starting at low population and getting up to a high population. And then you can relate that to the y-axis, which is the number of eggs per female. So what's the fecundity? So you can see that when numbers are low on the worms, females are very fertile, very fertile. But as we creep up to 20 and 30, those number of eggs per female drop drastically from, over 12 eggs to like 2 eggs per female worm, because it is density dependent. It is density dependent. The density of the worms affect the fertility of the females in that population. So this is absolutely a perfect example of density dependent. There could also be density independent population regulation. So that's when something outside of the population regulates it. So a natural disaster, for example, comes along and wipes out half the population. That is absolutely density independent population regulation. The number of animals had nothing to do with that natural disaster. K versus R selected species. So examples of K selected species. K selected species typically are going to have large, robust offspring. They're going to be living in a stable environment, a predictable environment are our K selected species. So if those babies make it, hopefully in this large, stable environment, they are going to have large adults just like themselves. But if we are talking about unstable habitats, where things are always constantly varying and always under threat, and that's the case, we call those guys our selected species, our selected species. So they're living in unstable, unpredictable environments with large numbers of offspring, self-sufficient offspring. So like we just talked about the dandelion, wind blows, we're going to throw seeds wherever we can get in the hopes that we're going to run the numbers and some of them will survive. Same with our little jellyfish over here. We're just going to release the offspring and the hopes that they're going to make it. They're going to make it. K versus R. Human population growth. Look at this J curve. Remember I was just talking about exponential growth. So we got 1,000 years ago, 1600s, 1700s, 1800s, and then look at it start to go. It starts to go. So we are starting to get modern agriculture, 1900, and then about 1950. 1950. You know what happened in about 1950? The first mass-produced antibiotics didn't come out until about 1950. So with modern agriculture, modern medicine, a whole bunch of stuff and other factors influence the human population. We have had exponential growth in the last century, 2 centuries. It's been crazy growth, crazy growth. So you can see them just going up. So the time between billions is this graph here. So from 1800 to 1930, we went from the global population from 1 billion to 2, from 1930 to 1960. You can see it jump from 2 to 3. And then we can see it keep continuing on and on. It took 130 years for the first billion. Then it only took 30 years to get to the second one, and then 15 years, and then 12 years. And it's getting smaller and smaller and smaller and smaller and smaller for us to increase our populations. So we're getting J curve exponential growth. So sometimes we are going to put that data, if we are a demographer and we're studying populations, we're going to put those data into age categories, and we're going to track those age categories and see how they do. So we might track them in an age structure diagram. So this is what we got here. Where is my mouse? There it is. So we got stage number 1, rapid growth, rapid growth. So everyone is trying to reproduce, and we are trying to get those babies to adulthood. And odds do not look that great because you can see it is very steep and very slim. Very few of this wide base made it to adulthood up here at 65. We've got slow growth over here to where we've had the same number of offspring, but it looks a little bit better. Like the numbers, it's more of a tapering instead of a J curve, a reverse J curve. Getting to puberty here on stage 2, it's slow and steady, more of like a 50-50 shot of getting to the next year. Remember how we talked about type 2 in birds? It's about a 50-50 shot on the slow growth to make it all the way up to adulthood. Stage 3, it's even better than 50-50. It is everyone in stage 3 is very, very stable. So almost nearly everyone that was born initially, almost, you can see it only curves just a little bit, made it to puberty. Almost everybody makes it to puberty in a stable population like this. And then you can see from age 15 to senior at 65, you can see that the population curves a little bit more and there's fewer there. But this is the textbook definition of stable. You can see as each age goes through, we have fewer and fewer, but it's shrinking at a gradual rate as it goes up. So this is a very stable population, is number 3. Number 1 is not a stable population. This is if you were living in a place like this is scary. This is scary in stage 1. Stage 3 is very, very stable. Stage 4 is kind of where some of our developed nations are currently. Because you can see age 15 was puberty, right? So there's more at 15 than there were being born. And at reproductive age, which technically is 15, I'm going to say it's higher. You can see that the adults in between 15 and 65 would be 50 years. So 25, 40. The midpoint here is 40. You can see that the people age 40 is about the highest level of population, and then it starts to shrink down. I would ask you, do you know why that is? But I'm just going to tell you. I'm just going to tell you. That's happening because the adults have decided to have fewer children, and not replace themselves, which is currently making the news because the United States is in a stage 4. It's in a stage 4. And so the population is not replacing itself. It is shrinking. It's shrinking. People are choosing not to have kids, and we could go on a whole soapbox about why that is, um, and the economy and how much things cost and healthcare and the medical field. But we're not gonna. I'm not gonna do it. But we could. So stage four is currently where we're heading now. Um, and if you're curious about your age bracket and comparing yourself to others, you could check out this link here for World Population Review. I think it's really cool. I was curious about my age cohort. I am late Gen X, at age 46. So you go in there and you plug in your age, and then you compare your age, versus the world, your age versus your birth cohort. So I could look at other Gen X in the United States, Gen X that were born in my age in 78 in other countries, of other continents. So I could compare births from 1978 to United States, births from 1978 to Africa, births from Gen X in Asia, Australia, South America, and you can check out your cohort and compare and see how it is. So it's pretty cool to see and compare. How many people are alive that are older than you, how many people are alive that are younger than you. It's just a neat little rabbit hole that you could go down if you're curious. Yeah, check it out. Check it out. Check out The World and Me, and play around in there. But you can see percent growth. We probably should be heading into pink instead of gray. I think United States is technically probably in the pink negative numbers now instead of the gray 0 to 1%, especially after COVID. All right, community ecology. Trying to keep track of time and make sure that we don't talk too much. On our community ecology, we've got a predator-prey dynamic graph, which I love to show, because when we talk about community, we talk about different populations, right? Different species, and we'll talk about when the community, different species interacting amongst themselves. And in this example, we got predators and prey. We have the hare, and we've got the lynx, and the lynx is going to feed on the hare. And excuse me, oof. You can see when the prey population goes up, then the lynx population follows a little bit later. If the prey population plummets down and there's no food to be had, then the predators are going to plummet down. So we can absolutely see those dynamics in this graph. Different types of uh, defenses that plants and animals may have. The locust tree absolutely has these gigantic thorns. Acacia trees, I tried to plant some native flowering trees when we bought my place, and it was from on Arbor Day, and Arbor Day, if and just in case y'all don't know, Arbor Day gives away free trees. I think it was a few weeks back, and they were giving out free trees in different areas. I know downtown Woodstock was giving away some free trees. They might still have some left, but I got a hawthorn, and I was like, oh, that's gonna be so pretty. It's got some lovely white blooms. It looks great. I planted it. It's got gigantic thorns on it. I should have known, judged by the name, that it would have thorns. I did not know that they were this big. They were this big. So I have them, they're still in the yard. But yeah, that is absolutely a herbivory defense. You don't want somebody eating on you, grow some thorns. Another defense, we got our turtles. Some of us got to play with turtles in class when we captured them out of the lake. And they looked a whole lot like this little guy right there. He's so cute. That's a little red-bellied, and our little pond sliders looked very similar. Eating, hide in a shell, that's a great defense. Other ones may be chemical defenses. So chemicals in all sorts of plants. We got a foxglove here, but there's tons of other plants out there with toxic properties that they can use to defend against herbivores coming along and trying to snatch them up. And then we've got other types of defenses. We've got this cute little millipede. He's adorable. And he will curl into the, little circles that they do. Some of those guys also have chemical defenses too. One of them, I believe, releases. It's in our area. He's kind of brightly colored. I've seen him on the trail a few times. They are like kind of yellow and red. And it releases a cyanide um, compound that is supposed to smell like almonds. And I would not touch him. I would not suggest picking him up because it's got mechanical and chemical defense. Body shape and coloration can absolutely be defense. So we've got this adorable little stick bug up here. I don't know if you've ever seen them, but they look like sticks and their behavior makes them walk like sticks. Like they've just got this kind of, looks like a branch in the breeze kind of movement every time they take a step. Same with the cutie patootie chameleons. They also kind of have that kind of goofy walk to them, but they are super cool because everybody knows chameleons can change their colors to match their environment. We don't have chameleons in our area. If you travel to Florida, they have lots of invasive chameleons down there. But we do have one that's native here. It's not a chameleon. It's the green anole. It's the green anole, and he is a cutie patootie. I hope y'all have seen them. You know what I'm talking about. But they'll change from green to brown, and the males will get this big old dewlap out when they're trying to find the females in the springtime, and they'll go out there and bring their little dewlap out and try and show off how big and bad they are. And they change color. So if they get really cold and they're inside in the wintertime, they're usually brown, but in the springtime, they will change to green. They'll go out and sit on the plants and be all pretty and green. Another defense with coloration is brightly colored or patterns to let predators know, don't mess with me. Don't mess with me. So we got the dart frog, the poison dart frog in the rainforest and absolutely brightly colored and totally toxic to predators. Everyone knows if you see the stripy squirrel, don't mess with skunks. Don't touch them because they may get you or your pet. And that's not a smell no anybody wants to play with. Another defense would be mimicry. Mimicry is super cool. We have the stinging insects out there, and then you'll have lots of flies that will mimic honeybees, and they'll look like honeybees, but they're not honeybees. But since they've got that pattern of yellow and black, everyone's gonna be like, "Oh no," and keep away and try not to mess with them because they might thinking that they're gonna sting them, and it's not. It's not. It's just a harmless little fly, going about being another adorable little pollinator out there, but he makes himself look like a bee. Butterflies. We have monarch butterflies that come through, and those guys, when they eat, they feed as caterpillars on milkweed, and that's really nasty and bitter tasting. And when they're adults, they're still nasty and bitter tasting. So when birds eat them, blah, they're gross and they spit them out. So there's other butterflies that will mimic that color. So the other birds won't eat them either because they'll think that they're nasty when really they're super delicious. But because they're colored that way, they get a pass. The competitive exclusion principle. With the competitive exclusion principle, it is going to be where you have two species that are fighting for the same resource. They're competing for the same resource, and not everybody's going to make it. Not everybody's going to do well if you're competing for the exact same resources. So we have paramecium. Paramecium were those cute little one-celled little guys that we saw that were covered in cilia. So we got paramecium aurelia is the green line, and you can see in 15 days its population skyrockets. This paramecium caudatum is a purple line, and you can see in 15 days its population gets pretty high too. If we put them both together in the same tube, in the same environment, you can see only one comes out on top. Only one is doing well. They had to compete for the same resources, and they did not do well together. You can see that Caudatum suffered and that they died off after 15, 16 days, but the Paramecium aurelia, they did really well. They outcompeted the Caudatum species. Another type of relationship is commensalism. So we're definitely gonna have questions about relationships. So commensalism is gonna be a relationship where one party benefits, and this is the bird example. The bird has built its nest in a tree. Now it's got shelter. The bird is benefiting, but the tree doesn't really care. The tree is not getting any sort of benefit from the bird being there, not really. So it could really care less. It could take it or leave it. And that's commensalism. One party is completely neutral, and then the other party gets a benefit, gets a positive. Symbiosis is a mutualism, symbiosis. So both parties benefit in symbiosis. And so we've got symbiosis in the ocean video, where I think in the past um, we talked about corals. We just talked about corals and coral bleaching events. And the corals are going to have their little symbiotic bacteria in there that photosynthesizes and produces sugars, but they have a safe place to stay inside the coral, and they both benefit. That's symbiosis, mutualism. Both parties are going to benefit. So mutualism example from the textbook is termites. They have gut bacteria, just like we do. We have gut bacteria too, and we've got a little mutualistic relationship with ours too. Their gut bacterium, they've got bacteria and they've also got archaea that live in their gut, and those both are going to help them break down wood. It's going to help them break down wood. So they can break down the cellulose where other creatures can't. That stuff would be, unsoluble fiber and it would just get away. But these guys can because they've got the gut biome to process it. So definitely mutualistic relationship with the bacteria, the archaea, the protozoans that live in their gut. Over here, we've got lichens. We mentioned lichens before, and those guys are going to have photosynthetic algae that live inside the little fungi sandwich. And we got the little algae that live in between there, like the peanut butter of our sandwich, on algae. So that's a mutualistic relationship for those little photosynthesizers inside of there, um, having a win-win relationship. Then another example of relationship is parasitism. So with parasitism, instead of both parties benefiting, only the parasite benefits, and the host isn't neutral. The host actually is negatively impacted with parasitism. So the example is tapeworm. We watched a video about tapeworms, and we all know to properly cook our meat and wash our hands, so we don't have to worry about this, hopefully. Moving on, we've got biodiversity, and that's going to talk about the number of species around the world. How many species do we have in an area? The species richness is our very first term up there. So species richness is going to be shown in this graph by the darker colors. So the darker colors you can see over here in the Amazon River Basin, this whole area, chock full, absolutely diverse, so rich in species. And we mentioned that when we learned about biomes in class too. We talked about different biomes. We said rainforest is crazy in species diversity versus desert. We talked about desert had extreme temperatures and it didn't have a whole lot of plants, and its species diversity was very low. So species richness very high, Amazon River Basin, maybe not so high up in the Arctic up there. Island biogeography. Island biogeography is talking about the uniqueness of islands. We were just talking about endemic species. We talked about how Australia and New Zealand have the kangaroos and the wallabies and the echidnas and those really cool animals that evolved on that island. And there's other places like that in these islands that have these special, unique creatures. So that is island biogeography, that they are isolated long enough that evolution happens and you get these really cool animals that live in places like Australia, New Zealand, Madagascar. Way back in the day, India used to be an island until it slammed up into the continent of Asia. So it's got some super cool creatures there too. So island biogeography is talking about Galapagos, how unique creatures can evolve on the islands. And then relative species abundance is just talking about the number of individuals of a species related to the number of individuals of all the species in the area. Okay, if we're going to describe species, sometimes species are described as foundation species. So an example in the coral reefs is absolutely you can't have a coral reef without its founder species, and those are the corals themselves, So the corals are going to provide the habitat, they're going to provide the food, the shelter for the other animals in the reef. So they're going to provide tons and tons of food and shelter, places to nest for the fish and eels and all the things, the shrimp, all the things that live on a coral reef, the vast array of species that live there can't be there without your foundation species of coral. Remember I was telling you about the video and I wanted to show you this video on TikTok and I couldn't find it. Here it is. It's Coral Vita. This is it. It's about 9 minutes long and it's talking about their hard work, about how they are working to repopulate the bleached reefs. Currently right now, in 2024, the Great Barrier Reef is experiencing its fifth bleaching event right now. right now of the Great Barrier Reef since 2016. So it had a huge mass bleaching event in 2016, and it is now on its fifth cycle of a mass bleaching event since then. So Coral Vita is doing stuff to fight that. So check out that video. It's really great. It's only 9 minutes long. Put it on like 1.5 speed, and you'll be through it in no time. But it's really cool. Keystone species, so foundational species, the building blocks of the environment. Keystone species. Keystone species are super important in its environment. Without them, you can see that they have a significant impact. They have a significant impact. If you remove the keystone, it is going to change immensely the species involved and how they interact in an environment. So in this example from the textbook, we've got Pisaster sea star, and it is a keystone species. So let's check out this little graph of this tidal pool. So in this tidal pool, we've got gooseneck barnacles and limpets and bivalves and acorn barnacles and little chitons, all of these species found in the tidal pool. And let's look at the number of species in this graph. So number of species present in 1963, in this tidal pool, we got all these species. And then we're going to follow the black line with the sea star in place. Excuse me, from 1963 to 1973, over 10 years, the number of species not only stayed pretty flat, but they also grew a bit. So the number of species diversified in 10 years in that tidal pool. But if we took out the sea star, if we remove the sea star, you can see the number of species on the red line plummets. It plummets. So in 3 years, without the sea star as a keystone species, the diversity just tanked. It just tanked. So this is an example of a keystone species is the sea star. At the very bottom, I've got my video about the gray wolf reintroduction into Yellowstone National Park. I love this video. It's great. It might exaggerate just a teensy bit, but it is an impactful, absolute keystone species in the area. So I do like to show the video of the impact of wolves. And I distinctly remember in the 90s when they did this, and it was hugely controversial. All the ranchers around Yellowstone were really, really lobbying hard to not have this happen, to not put the wolves back in there. And then they did put the wolves back in there. I think it was like 96, maybe 94 and 86, when they put the wolves back in Yellowstone, and they've been studying them ever since. And this video kind of tracks what happened when they put them back with the other species. Species that we do not want because they don't belong here are invasive species. And we call them invasive species because they don't belong. They come in and then they take over. They invade, they take over. They take over and they start using the natural resources, and they push out the local species to where they can't defend themselves against them. So we got lots of different examples of these. It's got purple loosestrife. We've got zebra mussels. I think those also came in from Mobile. I know we've got some of those in Mobile Bay watershed. We're known for zebra mussels and fire ants because we're a port, and all of those came in on ships. Garlic mustard. What else we got in here? The emerald ash borer. Yeah, that I think is still attacking the trees up in the Smokies. I think this little guy is still a problem up there. And then we got starlings. Starlings are definitely in our area. They are all over North America. They do not belong here. They were brought over here by the story goes, the urban legend goes that they were brought over by somebody who was a real big Shakespeare fan, wanted all the birds that Shakespeare discussed in his writings, and brought over starlings to have those birds over here too, because we did not have them, and then they took over. So lots of different examples of invasives. If you want to find out what's in the area, you can click on the database right there. If something happened in an environment and it was a devastating event, like a high intensity fire came through and wiped out everything, then you would study what happens in succession. How is this going to repopulate? So our very first stuff is called primary succession. So with primary succession, we've got some succulents. We've got some succulents growing in here, and it's Maui. It's Maui. I really want to go there. That's on my bucket list. Is to go check this stuff out. So we've got some primary succession. We got a lava flow. You can see all the lava underneath, and some of the first plants coming in there are these little succulents here. So this is our primary succession. And then in our textbook example over here, we've got sort of kind of a forest coming back. So we start off with our pioneer species, where we've got our kind of grasses and our shrubbies and our baby trees. And we got our intermediate species, where we've definitely got more bushes. The grass is still here at the bottom, but our trees are getting bigger and bigger. And then we got our climax community, where the trees are full grown. They are shading out the undergrowth underneath. And so we have fewer bushes and grasses in our climax community, because our overstory has shaded everything underneath. So we got secondary succession there. All right, behavior. This is the last bit of stuff, I think, is behavior. Right. Yes. Okay, so behavior, we have different types. On our innate behavior, I think we talked about some of this before, but innate behavior is stuff that you were born with. You just know how to do this. Like babies know how to suckle when they are born so they can nurse. It is an instinctive behavior. So out of the innate behaviors listed, we are discussing kinesis and taxis. So kinesis is movement. Kinesis is movement. And you have an example of these cute little pill bugs, these little roly-polies, or what I grew up calling them. And you can see the roly-polies in different environments and how it affects their movement. So in a moist environment, when it's wet, not so much. You can see there's not a movement. But if it's in a dry environment, they're going gangbusters. They're super excited. They're moving all over the place, and that affects their kinesis, their movement. Over here on the right-hand side is taxis, and that's going to be movement towards or away from something. Positive going towards something, negative taxis going away from something. And if we put the prefix in front of it, chemotaxis, we can see that this great white shark here followed the chum trail through the water, exhibiting positive chemotaxis to where it finally found the source of the bait here and attacked. So positive chemotaxis for the great white shark here. But there's also phototaxis when we talk about plants, right? Moving towards or away from the light source. Lots of our plants at home, if we don't move them, they are going to start to grow towards the window. Phototaxis, positive phototaxis. And then we talk about gravity, gravitropism, all that good stuff. Another behavior that we've got here, an example is a fixed action pattern, an FAP, fixed action pattern. And that is something that once it starts, we're going to do it. It's go time. And in this case, our example is the 3-spine stickleback. And this stickleback is a male stickleback. And when the male sticklebacks see red, literally like when they see the color red, they assume it's another male, and then they are going to fight it. They're going to fight it. So it's a fixed action pattern. If it is mating season and that fish sees another red color, it doesn't even have to be another fish. It just be the color red. It's going to set off their pattern. They're going to get all mad and aggressive because it's their fixed action pattern. If anybody has seen betta fish, they kind of do this too. If you put like a mirror up in front of them and they think that they're seeing themselves, but they think that they're seeing another male, they get all big and bad and their gills fluff up. Fixed action pattern. Another pattern is going to be migration. Migration, absolutely. So we got the great migration here of the wildebeest and all the things, all the things. So I got to go and see this 2 years ago, and it was fantastic. It was fantastic. And so everybody in the Masai Mara preserve, the animals in that valley in Kenya, they will follow the seasons. They will follow the seasons, so the dry season and the wet season, and they're going to follow the vegetation with the wet season, and they're going to follow the plants, the food source, and the predators follow the prey that migrate along. And it's just a great migration. They'll go from Kenya over to Tanzania and then back to Kenya. And you can see all these super cool animals out there just doing their thing, sourcing their food. Another thing is sourcing their food, foraging when they are foraging for food. So these are storks. We got some painted storks. If anybody has gone to the Atlanta Zoo, they used to have the flamingos, like kind of like right up front, and you could see them forage. But when they forage, they flip their heads upside down. And they kind of filter upside down. It's kind of neat to see. Speaking of birds and neat behaviors, courtship displays. I believe we did watch the lyrebird and how it was super awesome, and it could repeat like all these different crazy sounds. But if you did not see that, if you missed that class day, definitely check it out. It is fantastic. Another courtship display I've got here is the blue-footed booby, and it's little mating ritual trying to woo its female. Um, but that other video that I clicked that I told y'all about earlier, the whole movie, about I think it was why sex or something was what it was called. It has all of the mating displays in there, like so many of them. So those were courtship, love, displays of love, and now we have aggression, displays of aggression. If you saw a canine, a dog, coming up to you, ears pinned back, teeth on display, the haunches, the fur is all roughed up, its tail is down, and it is crouched. That dog is telling you with every bone in its body, without saying a single word, don't touch me. Not your friend. I'm not your friend. All the aggressive cues here just showing you. And then you hear the low growl and you know for sure, absolutely, not going to touch you, walking away. So aggression displays. Um, some are very, very obvious. Some are less obvious. My cat, for example, growing up was less obvious. He would look at you and you'd be doing nothing. You'd just be sitting there watching TV and he'd just get mad. And the only giveaway was his pupils would get really, really big, and that would be the giveaway that he's in fight or flight. Because sometimes the ears would be back, sometimes they wouldn't. Sometimes the tail would twitch, sometimes it wouldn't. And then just so if you can read the body language, it helps. It helps to figure out if they are going into aggressive mode. Another type of behavior, distraction. Killdeer. We used to have these a lot on campus, way before years ago. We didn't always mow that piece of land between the main campus and the baseball field and softball field. That didn't used to always get mowed, and it used to grow up pretty tall, like several feet tall, with weeds. And remember, we just talked about primary succession. There was high grass, there were bushes in there, and there were like trees, baby trees growing in that field. And we would have killdeer that would go and nest in there, and deer would come and sleep in there and nap in there. Coyotes would hunt in there. But sometimes you used to be able to go and see them. You could see the killdeer. So when they were nesting and they had their little babies and their little fluffers would hatch, Mama does this distraction technique. So I got a link to it here, and you can see she pretends like her wing's broken if predators come along. So if a hawk is around, then she's going to wander away from her babies, pretend like she's got a broken wing, distract the predator over there so her babies can run and get someplace safe. And then, ooh, magic, her wing is all fixed up and she can escape. Um, selection. So we got intersexual selection and we have intrasexual, God, I can't speak tonight. Intrasexual selection. And so with intra is going to be where we are competing within within ourselves. So we are fighting the same sex. We are fighting, example, males going against other males competing for their mates. Deer are my go-to example. I think elk may be in the picture. Yeah. Got a picture of an elk here. They're absolutely going to compete, male versus male, intrasexual sexual selection. Then we got intersexual selection, um, going after the other sex, trying to find the mate. Here's my Y sex video right here. We've got the whole hour-long video. Um, so we are trying to compete and find a mate. We're trying to compete against our same sex for mates. Sometimes the relationship is monogamous. Sometimes we're monogamous for just the season. Sometimes we're not even monogamous for that. And sometimes there's polygynous and polyandrous. So monogamous, supposedly mate for life, with our cute little penguins there, that aren't so little. Those guys are really big, actually. Then we've got elk, polygamous, where they've got many females and one male, in his harem or his herd. Polyandry, where we've got one male, one female, and several males. So with these guys, the males raise the young, and so the female will come along and lay her eggs, and he'll fertilize the eggs and raise little babies in his little pouch. This is polyandry's opposite. Then we've got different types of behavior. We've got innate that we told you that you were born with. These are instincts you just know. And then we've got learned behavior, learned behavior. So habituation is definitely learned behavior. I was telling y'all about working with bears and how you didn't want to get bears habituated to people. That's bad. We don't want bears thinking that, well, Susie leaves her dog bowl outside with her dog food in it, and there's always bird seed in the bird feeder. So I'm going to come to Susie's house every day and get that food. And Susie eventually isn't going to mind. It's going to be fine. And so when then Susie's not there, I'm going to go over next door and go to Rob's house and check out his house because Susie today didn't have food and people are fine. It's no big deal. I'll just go bust into Rob's kitchen. No, we don't want to habituate wild animals. The example here in the textbook is pigeons. Most of us have probably been in the Walmart parking lot at some point, and there's usually some little songbirds out there that are habituated and will move at the very last second, but people are not going to bother them, just like these pigeons in this picture. Another type of behavior is imprinting. We've got an example of ducks here. I've got an example of raising cranes because these cranes were endangered and they still really are. Their population suffered a lot. But when they are born, they're going to imprint on one of the first things that they see, and that's you. Whatever I see is my parent. That's my parent. And so if you were raising endangered birds yourself, and that's the first thing they see. They think you're the parent. So eventually they figured out that imprinting was a thing and that when they were trying to raise these crane chicks, you can see in the video that they are dressed like cranes. It looks ridiculous, but they're trying to teach them that this is what an adult looks like, not a human. Don't associate people as your parents. Um, and it's a whole story there, but you can see an example of them trying to do the right thing and get these cranes raised up and out to adulthood. Then we got our classical conditioning, Pavlov's dog. So the story goes that Pavlov would feed his dog, and every time he fed his dog, he would ring a bell. He would ring a bell. And I don't know if your dog does this. My dog definitely does this, but my dog, when she's hungry, she drools. She drools. And same with Pavlov's dog. Pavlov's dog would drool in preparation for that tasty, delicious meal. So Pavlov did this classical conditioning. Ring the bell as the dog's eating. So bell means food. Bell means food. And eventually you ring the bell, dog starts salivating, even if there's no food, even if there's no food. So that's Pavlov's dog. And I've got the Office example. I don't know if y'all have seen this. I don't know if y'all are Office fans, but it's cute and it's a great example of classical conditioning. Operant conditioning is where the animal is going to do a type of behavior, and that type of behavior is going to be hopefully rewarded. Hopefully, we are doing positive reinforcement of that behavior. So here we do have an example of positive reinforcement. We have some dolphins that are doing behavior they didn't normally do in the wild, and when they do that behavior, they get some fish. They get a positive reinforcement for that. There are other examples of um, things being done to rats in cages and electrocution and negative reinforcement for this button. So this button gives you treats and this button is going to shock you. There's some stuff out there. But if you are getting a positive or negative experience for a type of behavior, that is operant conditioning. Food rewards, speaking of positive conditioning and positive rewards, food rewards, there are three different lines over here on this graph, and you can see literal rat race. It's a rat race. So we got the start, and here's a finish over here. And so with group one, the green line, sorry, with the green line, our little rodent friends, you can see they started with 6 errors on day 1 to make it to the end. It took them 6 different mistakes to finally make it to the end. By the time that they made it to the end, they had 6 mistakes. But you can see by day 5 that drops down. That drops down because at the end they're getting food. So they got more incentive to get to the food faster, and they're making it with less errors. So you can see the number of errors drops by day 5. And this is the first group. Group 1 got food immediately on completion of the maze. Group 3 got food on day 3, right? Group 3 was day 3. Yeah, so in the first 3 days, no food reward, but on day 3 and after, food reward. So you can see it kind of flat lines there from 6 to 5, but it's still pretty stagnant. But on day 3, 4, 5, we figured it out. We figured it out and those errors dropped down because food was definitely a motivating factor. You can see the one in group 2 is the blue dotted line. They didn't get food until day 6, and you can see their number of errors did not improve pretty much throughout the whole study until 2 weeks in. Until 2 weeks in, they didn't get a food reward until the first 6 days. So it makes a difference. It makes a difference. I myself am food motivated too. All right, so that's the end of lecture. Hope I didn't talk for too long. If you have any questions, I will see y'all in class. You can shoot me an email. Ta-ta for now. Bye.